I'm a Maize researcher. I worked with Nathan Springer. Those of you who know him know he works in Maize. And so when I started to work at this Hamline University, a small school, liberal arts school, almost no research capabilities, I brought my research project in there. And so what I want to tell you today about is how I'm trying to engage my students, both research students, but primarily for today's talk, my students in my introductory genetics class. And many of those research type investigations that are being developed and run are targeted primarily to upper level major students. And that's reasonable, right? Those are students that have some interest in the field, they have some knowledge, some prior knowledge from their prior courses. But it's really important for introductory major students, but also for non-major students, to have similar types of experiences, so that they know what to expect, so that they know they could get really interested in your field and get into your biology upper level classes. So I'm trying to use these types of real research experiences into my introductory courses to bring students into my lab to do research with me and into my upper level courses. Some of my students really didn't realize that biology and data science are in any way connected, so they don't really see biology as a big data science. And a lot of surveys find that our biology students don't have those tools, and I can for sure speak for my students. My students, sometimes many of them come to biology because it's not math. So I want to tell you more about my project specifically and what I was doing with the students. I'm sure, I know you've seen this picture more. Part of the things that I'm doing with my students is about these green lines that you had sort of hiding before in some of those pictures of DNA subway. But this is a line that has been developed as the latest, and it uses iPlant discovery environment, essentially, programs to help students analyze those large data sets from RNA sequence analysis. And so I will tell you a little bit about how we use this. What I'm interested in is looking at Maize and looking at how Maize reacts to stress. One of the challenges I had with recruiting my students to my labs is that most of our bright students want to work in medicine. They don't, I tell them I work on Maize, they're like, okay, whatever, I'm not interested. I am not into plants, I want to work with an immunology professor. I want to solve the whole cancer issue. I don't want to work with you. Many of my students are first generation students as well, and they have farming families, so they know that those farmers, their parents, their grandparents, they can't plant corn in Minnesota early. They want to, but they can't because all of those cold snaps are gonna, harm. And so this worked well as my hook to bring my students, to get them interested, is that, okay, we have this environment changes, we really need to try to figure out how Maize responds to cold and whether we can actually do something about it. But it's essentially an introductory course. I had 100 students, 5 lab sections. I teach all of the lab sections, so I could really work with the students during the labs doing these types of analysis. And so they worked in teams. They did a lot of different inquiry-based active learning exercises, learning about RNA-seq analysis. Students can actually ask interesting questions. If I would be doing this research, I would also be interested in asking some of those types of questions. And that's where green line came in, is that you can put the data into the green line, you can make those projects in there, and then students can look at the quality of the data, for example. So the pictures are generated, students can generate those types of pictures, and this is an example of a relatively good quality run where most of the sequences are in this green area and not too many are in the red area. Another thing that I really liked about this is that students get to see how the data actually looks like. And I really had to spend a lot of time encouraging them. I really had to spend a lot of time telling that, hey, when I started this project, that's how I looked like. That's how confused I was. I didn't know how to do this, and that's okay. And at that point, the students, when they ran some of this analysis and they realized how differentially, how many genes are differentially expressed, they were like, what's going on? My hypothesis was 10 genes. Now I'm getting, tens of thousands of genes. I did something wrong, and I really had to encourage them, no, you're okay, that's fine that your hypothesis is wrong, that's fine. It doesn't mean that you failed, it means you found something you didn't know before.